Hi, I'm Rob Kirkpatrick, and I'm here to say a few words about multi-trait genetic analysis. Specifically, we'll talk about two classes of theoretical models, the independent pathway and common pathway models. These are said to be theoretical because they can be used to represent some substantive theory you might have about the decomposition of variants common to multiple traits, as well as unique to each trait. The common pathway model and the independent pathway model are both extensions of the generic common factor model, which is depicted here. Under the common factor model, one or more latent variables, known as common factors, are hypothesized to underlie variance that is common to more than one trait. In the diagram here, there is just one common factor, F1. The observed phenotypes are represented by P1 through P4. Each phenotype has a loading, which is its coefficient of regression onto F1. F1's variance is fixed at 1.0. But that's not the whole story of the common factor model. Each phenotype will also have residual variance that is left over from its regression onto the common factor. In orthodox psychometric terminology, this residual variance is the phenotype's unique variance and is represented here by four unique factors, one for each phenotype, E1 through E4. Under the assumptions of the common factor model, the unique factors are uncorrelated with one another. In this diagram, each of the four phenotypes is regressed onto its corresponding unique factor, and the unique factors have their variances fixed to 1.0. If we consider our unit of analysis to be the twin pair instead of the individual person, we then have the same common factor structure present twice in our structural model, once for each twin. The twins' common factors are correlated to some degree, which depends, among other things, upon their zygosity and whether they were reared together or apart. The matrix on the right-hand side of the first equation represents variance-covariance common to more than one phenotype. It is a function of the phenotype's loadings onto the common factor. The matrix on the right-hand side of the second equation represents variance unique to each phenotype. It is a function of the phenotype's loadings onto their unique factors. This slide introduces two new things. One is that a triangle with a 1 in it that has one-headed paths going from it to all the phenotypes labeled mu1 through mu4. That just represents the phenotypic means in this diagram. The more important new thing is the addition of a common additive genetic factor. We can also add a common shared environment factor and a common non-shared environment factor. Those three common factors respectively represent additive genetic, shared environmental, and non-shared environmental variance that is common to more than one phenotype. We can also biometrically decompose each phenotype's unique variance. For instance, AS1, CS1, and ES1 respectively represent additive genetic, shared environmental, and non-shared environmental variance that is unique to P1. The situation with the common A, C, and E factors is analogous to that of the generic common factor model. The matrix on the right-hand side of the first equation is the common additive genetic covariance matrix, whereas the matrix on the right-hand side of the second equation is the unique additive genetic covariance matrix. There are, likewise, common and unique shared and non-shared environmental covariance matrices. The bottom of the slide presents some OpenMx syntax for constructing the two loadings matrices. The matrix of loadings onto the common additive genetic factor has R symbol path AC and MX name AC. Now, by MX name, I refer to what the object will be called within the MX model's namespace. The matrix of loadings onto the unique additive genetic factors has R symbol path AS and MX name AS. Total additive genetic covariance in the phenotypes is the sum of common and unique additive genetic covariance matrices. The total additive genetic covariance matrix has R symbol cov A and MX name A. The bottom of the slide presents OpenMx syntax for constructing the total additive genetic covariance matrix. The model that we have just described is known as the independent pathway model. It is sometimes referred to as a biometric factor model. It allows for different covariance structures for common A, C, and E variants. We have so far examined merely the simplest case of the independent pathway model. This slide depicts one possible form of the independent pathway model for 6 phenotypes. Of course, it is additionally possible to biometrically decompose the residual or unique variants of phenotypes. The table in this slide presents the MX names of the loadings matrices for the common factors and the unique factors. Here, NV means number of variables, that is, the number of phenotypes. The loadings matrices for the common factors are NV length column vectors. The loadings matrices for the residual or unique factors are NV by NV diagonal matrices. The independent pathway model can be extended to have more than one A, C, and E common factor. But be careful when adding common factors to the model. Total parameters per source of variance cannot exceed NV times the quantity NV plus 1, whole thing divided by 2, where again, NV refers to the number of phenotypes. Also, for a common factor that has only 2 indicators, the 2 factor loadings on the latent factor need to be equated, or instead, a correlation could be estimated between the 2 residual factors or unique factors of the same source of variance on those 2 indicators. If you have more than one A, C, or E common factor, your syntax that constructs the corresponding loadings matrix needs to accommodate that. In general, the corresponding common factor loadings matrix will be NV by NF, where NF is the number of common factors of that type, A, C, or E, as the case may be. The loadings matrix for the unique factors will pretty much always be an NV by NV diagonal matrix. In any event, the total additive genetic covariance matrix will always be the sum of the common and unique additive genetic covariance matrices, with the analogous case holding true for the shared and non-shared environment. This slide just contains a few pointers about syntax. For one, it can be helpful to put all relevant matrices together into a list, which can be concisely used and reused to construct MX model objects. In this syntax, the list has R symbol pars, which is used when constructing the MX model objects for the MZ twin and DZ twin groups. The group MX models are in turn used to construct a container MX model, and that is the MX model object that is actually run. For your reference, this slide just shows the structure of the loadings matrices and the labels of the free parameters they contain. And as you'll see in the script, you can compare the independent pathway model to an atheoretical model, which can use either the Koleski parameterization or the direct symmetric parameterization. You can also obtain standardized estimates of the loadings as shown here. To be clear, these are the coefficients of the one-headed paths going from latent factors to observed phenotypes, assuming the variables on both ends of the paths have a variance of 1.0. This slide depicts a path diagram for an independent pathway model that has not 1, but 3 common additive genetic factors. Now, for ease of presentation, the environmental factors are not shown. This kind of factor structure is called a bifactor model because there are 2 types of common factors at the same level of hierarchy. One type is the general factor, which has paths going to all the phenotypes. In our case, A1 is the general factor. The other type is the group factor, which has paths going to some but not all of the phenotypes. In our case, A2 and A3 are group factors. Note that all 3 factors are mutually uncorrelated. If you want to fit the additive genetic bifactor model we just saw, you need only add 3 new lines of syntax and only modify 1 line in the base script. Okay. Let's now change gears and return to the generic common factor model we covered at the beginning of this slide deck. We can think of the common factor as a latent phenotype or latent trait. And as such, we can biometrically decompose variance in the latent trait into additive genetic, shared environmental, and non-shared environmental components. The latent factors AL1, CL1, and EL1 that you see here are examples of higher order factors. And as before, we can biometrically decompose each phenotype's specific or unique variance. This kind of model doesn't introduce anything to the structure of the unique variances that we haven't seen before. What's new is the structure for the common variance. The matrix on the right-hand side of the first equation is the common additive genetic covariance matrix. It is a function of the phenotype's loadings as well as the latent traits loading onto the higher order additive genetic factor. The bottom of the slide presents some OpenMx syntax for constructing the two loadings matrices. The matrix of loadings onto the latent trait has R symbol path FC and MX name FL. The latent traits loading onto its additive genetic factor has R symbol path AL and MX name AL. As before, total additive genetic covariance in the phenotypes is the sum of the common and unique additive genetic covariance matrices. The total additive genetic covariance matrix has R symbol cov A and MX name A. The bottom of this slide presents OpenMx syntax for constructing the total additive genetic covariance matrix. The model that we have just described is known as the common pathway model. It is sometimes referred to as a psychometric factor model. Under the common pathway model, common A, C, and E variants are each represented by a component of variance in the latent trait. Thus, the model predicts the same covariance structure for A, C, and E. This slide depicts one possible form of the common pathway model for 6 phenotypes. Of course, it is additionally possible to biometrically decompose the residual or unique variants of phenotypes. The table in this slide presents the MX names of the loadings matrices for the common factors and unique factors. NV again means number of variables, that is the number of phenotypes. The phenotype's matrix of loadings onto the latent trait is an NV length column vector, whereas the latent trait's loadings onto the higher order A, C, and E factors are scalars. As in the independent pathway model, the loadings matrices for the residual or unique factors are again NV by NV diagonal matrices. Okay, the important takeaway from this slide is that we need to impose some kind of constraint on this model for purposes of model identification. Here we have decided to constrain the variance of the latent trait to 1.0. As before, it can be helpful to put all relevant matrices together into a list, which can be concisely used and reused to construct MX model objects. However, the MX constraint object should only go into one MX model object, which in this case is the container MX model object. For your reference, this slide just shows the structure of the loadings matrices and the labels of the free parameters they contain. You can also obtain standardized estimates of the path coefficients as shown here. To be clear, again, these are the coefficients of the one-headed paths, assuming the variables on both ends of the paths have a variance of 1.0. This slide depicts a path diagram for a common pathway model that has not 1, but 3 latent traits. This is another instance of a bi-factor model. Here, L1 is the general factor, whereas L2 and L3 are the group factors. And again, as in the case of the common pathway model with just one latent trait, you can inspect the standardized path coefficients of the bi-factor common pathway model, which are presented here. And finally, you can use MX compare to compare your independent pathway and common pathway models to your atheoretical model, whether your atheoretical model uses the Koleski or direct symmetric parameterization.